I'm your host, Adrian. We do content like this weekly, so hit the subscribe button and ring the bell to get notified. If you want more content like this, click the like button and please leave a comment. Thanks again for watching.
Welcome to today's Rupa University Lab class presented by Rupa Health, the simplest way to order specialty lab work. My name is Adrian Martinez and I will be your host for this afternoon. Today we have a very, very special guest in Roger Deutsch here to discuss balancing innate and adaptive immunity, specifically with nutrition-focused testing. Before jumping in, a couple of quick housekeeping items. Everyone joining will be muted by default, but don't fret. If you have any questions, feel free to use that Q&A button down in the right side of your menu, and we'll host a live Q&A session at the end of the presentation. Immediately following the Q&A, yours truly will be doing a quick walkthrough of Rupa Health, chatting about who we are, what we do, why we're so passionate about functional medicine, as well as showing you how you can order today's test. If you have to jump early, no worries at all. We'll be sending out a recording of today's session along with the slides within the next few days, so no worries there. But with that, let's go ahead and jump in. I'd like to introduce Roger Deutsch. He is the CEO and founder of Cell Science Systems. For over 35 years, he's been active in numerous research projects involving cellular testing for sensitivities and nutritional assessment. He's a frequent lecturer and co-author of Your Hidden Food Allergies Are Making You Fat. With that, I'd like to hand the reins over to Roger. Thank you so much for joining us today. I'll let you take it from here.
Thanks very much, Adrian, and good morning to everyone, or good afternoon, as the case may be. So today I will be speaking about how to balance, or how nutrition can be used and diet can be used to balance innate and adaptive immunity, and particularly why we're interested in doing that. And let's get this show going. Some of our collaborations, we've had some research grants from government and some private industry grants to do research, particularly on ALCAT. Today's topic is how to balance innate and adaptive immunity, and we're going to look at it from two perspectives. First of all, this is the cover of the January 13th issue of Science magazine, which is a very mainstream magazine. And most of you, a scientific journal, it's been around since the 1800s, and most of you will recognize that this is an immune cell, specifically a lymphocyte, with a yin-yang sign superimposed on top of it. And the whole issue is dedicated to inflammation and the dangers of not resolving inflammation after it's accomplished what it was meant to accomplish. So the implication here is that there needs to be a balance. And that's exactly what we're going to talk about between the innate and adaptive immune system. We're going to speak a little bit about what those are. And within those systems, between activation and rest and resolution. So the key points are as follows. Chronic activation of innate immunity underlies degenerative disease as well as the cytokine storm. And I will be speaking a little bit about COVID because it's so important. It's all related to the immune system function. It's upended everybody's life on the planet nearly. And the outcomes depend upon our immune function, the strength of our immune system, and the overall state of restfulness of the immune system. And what some people don't necessarily know is that the innate immune system is responsible for the cytokine storm. And it also suppresses specific immune cell function. And the things that trigger the immune system, of course, are the obvious things, what we call pathogen-associated molecular patterns. But also there are sterile triggers. And that's what we're going to focus on. Those include things like chemicals, toxins, cellular debris, which includes cell-free DNA, we're going to focus on a bit, which activate what we call DAMPs, which are danger-associated molecular patterns. They may be sterile, but they can activate immunity. Cell-free DNA is an area of very active scientific investigation right now. We're going to focus on that, and particularly in its relationship to our food sensitivity testing called the ALCAT test. And functional cellular responses that identify sterile triggers are the tests we're going to be speaking about, the ALCAT test, as well as tests that guide diet and supplementation. to strengthen the other branch of the immune system, the specific immune response, which are a group of tests we call cellular nutrition assays. So COVID, as we can well appreciate, has outcomes ranging from being totally asymptomatic to being fatal. And some of the factors that influence that are, well, most of the factors have to do, let me rephrase that, all the factors have to do with how our immune system functions. So people with underlying conditions and elderly who obviously have compromised, may have compromised immune systems, not necessarily, but people with conditions like asthma, obesity, cardiovascular disease, all things that stem from chronic activation of the innate immune system are more likely to have a worse outcome, but also gender. So the purpose of showing this slide is to show that gender has a big role to play, and females are 40% less likely or more likely to survive than males. So it proves once again that women are actually smarter than men and their immune systems are smarter. So the innate immune system pathways, and this is a publication from Science Immunology just a few weeks ago, talk about neutrophil extracellular traps, or NETs, which of course are one of the major defenses of the innate immune system. They are found in the lungs of patients with COVID. And they have been reported to induce apoptosis or death of lung tissue and to contribute also to thrombosis, immunothrombosis. Not only that, these innate immune cells will suppress T cell induced interferon gamma production, which of course is a major benefit to countering the virus or other infections. In this illustration from that paper, we see the three situations over on the far left, the normal situation where there are immune cells in the alveolar of the lung tissue, which are there surveilling. In a mild case, there is some activation. There's dendritic cell activation, there's macrophage activation, there's neutrophil activation, there's secretion of toxic chemicals to destroy the pathogen. When it gets out of hand, then we get this going further, and then the granulocytes, the neutrophils, produce those nets, those traps. And they also cause tissue damage, which create damps, which further creates a vicious cycle. And the cells are also producing a lot of free radicals. Then there's damage to the endothelium of the capillaries that are nearby. There's a normal gap normally. A fluid comes in and we have all the sequelae of a serious COVID situation. So it's a question of having an appropriate immune response or an exaggerated immune response. And in this paper, it is indicated that a Western diet can trigger an exaggerated immune response by reprogramming the innate immune cells such that they train the immune system in such fashion that when there's a secondary stimulus, such as a damp, there will be an exaggerated response and potentially pathological outcomes. So it's nice that mainstream medicine, this is published in Cell, which is one of our more well-respected journals, indicates that Western diet is not healthy, which of course is not a surprise to anyone on this call. And chronic activation of the innate immune system suppresses specific immunity. So this is another paper from a respected journal, Atherosclerosis. And what's interesting is that the authors here did a meta-analysis of 8 other studies, looking at simply the ratio of neutrophils, which are the predominant innate immune cells, to lymphocytes. And they found that the people who had the higher ratio, more lymphocytes, more neutrophils, fewer lymphocytes, had a much greater, as an average of the all 8 studies, 2.33 times odds ratio of having a cardiovascular event. Of having all-cause mortality as well as fatality. And the relative risk of cardiovascular events was almost double in those people who had the lowest quartile, the highest quartile versus the lowest quartile of neutrophils to lymphocytes. So again, here's a question of balance. It's clear illustration that in modern times, we have more neutrophil activation because we have more sterile insults from the environment, and neutrophils become activated and they die. And we'll talk about that. They die in different ways. And new neutrophils are constantly generated to replace those. And when there's an imbalance, as we see here, between those type of cells and the lymphocytes, which are more specialized, it's very, very unhealthy. So, moving onwards. Why does it happen? So here's a paper I looked at recently, just last month, from Nature Aging. And it's kind of interesting in that they found out that with older subjects receiving a vaccination shot, just the trauma of receiving the shot, the injection, was enough to induce senescent fibroblasts to produce chemokines and express chemokines, which would upregulate innate immunity and then suppress specific immunity. And they go into what the factors are, and I delve deeply into it. Interesting though, it wasn't all the older subjects that experienced this. And of course, they included control groups, younger folks, but it was the one who had background inflammation. So there was a group of about 20 subjects over the age of 70 who didn't have this phenomenon. So it is possible to prevent some of the dangers of the normal aging process that occurs and its impact on the immune system. So the results point to a series of molecular and cellular mechanisms, which is very interesting to review, which you can do later, that induce cellular senescence and link cellular senescence and tissue damage. And senescent cells, by the way, of course, are obviously a damp, causing tissue damage, excess inflammation, reduced immune responsiveness in the skin. But they also believe that that relates to other tissues and organs, and it can be inhibited. They found that by using anti-inflammatories, you can abrogate that response. In another paper from the same journal, also recently, about 6 months ago, a group of investigators from Stanford and the Buck Institute of Aging reported upon a way to assess frailty and cardiovascular aging. And what it involves is the ability of the specific immune system to respond to stimuli. So chronic and systemic inflammation is thought to be triggered by physical, chemical, or metabolic stimuli, sterile agents, such as those released by damaged cells and environmental insults, termed DAMPs. It's associated with aging, is characterized by low-grade and persistent inflammation, ultimately leading to collateral damage to tissues and organs. So let's begin to focus a little bit on what is probably one of the major damps that we're dealing with. In this paper, Frontiers in Bioscience, a few years ago, a group of traumatologists expressed their view about circulating or cell-free mitochondrial DNA, and they made an interesting statement. They believe that our current knowledge of cell-free circulating DNA is rather exploratory, with the potential to rewrite in the future, to rewrite the pathology of the leading causes of morbidity and mortality, such as inflammatory conditions, autoimmunity, cancer, heart disease, stroke, and injury. So they're linking here cell-free DNA, particularly mitochondrial DNA. Of course, we have DNA in the nuclei. We have mitochondrial DNA, and that drives the mitochondria. And when a cell undergoes certain kind of processes, which we'll talk about, it can be released, and it can drive these factors, these conditions. So this is actually a micrograph of an innate immune cell, in this case a neutrophil, which is undergoing the process of mitosis. Neutrophil extracellular traps. So in this process, the cell actually enlarges about 16 times and extrudes contents from within, which are comprised of certainly toxic enzymes, lysozyme and myeloperoxidase, calprotectin, elastase, and contents of the nucleus, the chromatin, which is comprised of the DNA, the nuclear DNA, and the histones, which are the proteins within the nucleosome of the nucleus that the DNA is wrapped around to create order. But certain enzymes, when there's an infection or another trigger, which could be a sterile trigger that activates the innate immune cells, occurs, there will be processes, the decondensation of the chromatin, meaning the DNA material and the histones, and the other toxic components, as well as the cell membranes and other proteins. And they form these traps, and they'll trap, as in this micrograph, bacteria. And it's trapped by membrane material, but it's then destroyed by the toxicity of the DNA and histones, which are actually quite toxic. Now, hopefully, after this process occurs, it's then resolved. In other words, phagocytes come in and remove all the debris, and life goes on. But when there's too much of it, or if someone suffers from a deficiency in the enzymes that degrade the DNA or the RNA, which are DNases, RNases, respectively, there can be too much of this stuff. Or if the system is overloaded, if we're in a toxic environment and the immune system is chronically activated, it can overwhelm these systems, and we have too much of this, and it produces the chronic activation of the system. So maybe for some of you, this is the first time you're hearing about NETs, but I can assure you that since their discovery in 2004, there's been a steep slope in terms of the number of research papers that have come out looking at NETs. So I updated a little bit of an observation of those papers recently on PubMed that have to do with NETs. And you can see from just a few weeks ago, the number of papers on PubMed having to do with NETs and inflammation was over 1,200. With COVID complications was 55. With COVID fatality, 14. NETs and thrombosis, 56. NETs and autoimmunity, 469. NETs and cardiovascular disease, 700 plus. NETs and cancer, close to 3,500. NETs and aging, close to 200. NETs and dementia, close to 70. It goes on, respiratory disease, 670. Pneumonia, 270. Organ failure, obesity, eye disease, liver disease, and cystic fibrosis. In fact, the one drug that's on the market now for dealing with cystic fibrosis is aimed at, it uses DNases to degrade NETs in the lungs. It's inhaled. So one of those papers, for example, showed that inflammatory microvascular thrombi are present in the lung, kidney, and heart. I'm sure it extends to other tissue as well, and contain neutrophil extracellular traps associated with platelets and fibrin. So this is a relevant factor today. The dark side of NETs was described as being NETs contain neoantigens, some of which are modified histones. Again, histones are the proteins or proteins, and there's a bunch of them in the nucleosome. The DNA is wrapped around that. And why? The histones have a positive electrical charge. The DNA has a negative charge. So it keeps it tightly grouped there. Acetylation of part of the histones, a lysine tail, can change that and then loosen up the DNA in a certain area, and that gene can be expressed. So it's one of the key factors of epigenetics. But those things are toxic, and when that breakdown happens and those histone proteins go out in a modified form, they act as neoantigens, and they damage tissue anywhere, and then they can spur or initiate the formation of autoantibodies against that tissue. So NETs have also been, as shown in the previous slide, associated with quite a bit of autoimmune disorders, particularly lupus and arthritis. And just an illustration of how that affects different tissue, the arteries, the joints, the cardiovascular, the vessels, the NETs combined with, for example, myeloperoxidase or other toxic enzymes, anti-nuclear cytosolic antibodies, citrulline-aided histones causing arthritis, and NETs combining with other factors, platelets and fibrin and MPO and so forth, and causing damage or thrombi. So this is a feature of COVID, and it's a feature of aging. And if we can control it, I think we can prevent the bad aspects of both of those. So just to shift right now, I've got a good sense, I think, from this, what the scope of the problem is. And I think it's important, we understand that, and I think we do, that the condition of the immune system is absolutely foundational and critical to everything, including these challenges we see today that bring into focus what the weaknesses are in the organism and what it's drilling down and identifying is immune function and homeostasis and balance. So several years ago, we've always been very open and transparent about collaborating with academic institutions, with our universities. Some years ago, people from Yale School of Medicine had been involved in an NIH-funded research project evaluating the use of non-conventional medical tests. And from that, they found a lot of people were favoring, doing a lot of food sensitivity testing and favoring ALCAT. And so they wanted to perform a study. We agreed. And first thing they looked at was IBS, because one of the principal investigators was a gastroenterologist as well as an immunologist. And so to make a short analysis here, this was a randomized control, placebo-controlled trial looking at the benefits of an ALCAT-generated diet in patients who had irritable bowel syndrome. And the endpoints were, the two major endpoints were symptom scores, general improvement score, or global improvement score, and symptom severity score. And the blue line in this case is the change in symptoms in the treatment group, and the kind of orangey, rust-colored line is over a period of 8 weeks, same period, the control group. And they both followed an ALCAT diet, but the therapeutic group followed an ALCAT diet that caused them to eliminate the ALCAT positive foods. The control group followed a sham diet, which was based on ALCAT, but kept in their reactive foods, but took out foods from the same category in the same number. So if they were reactive to 2 meats and 2 vegetables and 2 fruit, they would eliminate 2 meats, 2 vegetables, and 2 fruit, but not the same ones as the test indicated. So in this way, we had a really good control. So one can't say that, well, this group just eliminated all their wheat and dairy and this group kept it in. No, it was totally randomized and controlled. And the differences you can see are quite dramatic, especially after 8 weeks. There is a placebo effect. Anytime people change their diet, they're going to get better. There was probably some benefit from removing foods of the same group that were test positive, but still the people who followed the ALCAT had a better outcome by at least 100% or more. So this led to some further research from that group. Again, one of the folks was an immunologist. So they included a broad spectrum. They looked at the people who had the best outcomes. There were 59 people in the test, and then they looked retrospectively at 8 of them who had the best outcomes, and they did an in-depth analysis of over 1,300 proteins in the plasma pre-therapy and post-therapy. And there were a number of interesting changes in various proteins in the serum, but one that kind of stands out is the elastase, neutrophil elastase, which will degrade tissues, and it will actually degrade the gut barrier and cause leaky gut. So, of course, it's a major component in NETs as well. So it was interesting to see sort of a cross-platform proteinomic validation of what the cellular testing was indicated. And one of the other key findings was that when you looked at an ALCAT positive reaction, and this was published in a separate study, when you looked at an ALCAT positive reaction, there was a greater amount of cell-free DNA found in the supernatant following an ALCAT food exposure versus a ALCAT positive food versus an ALCAT negative food. So that kind of was an interesting observation. And that led to some further research from some other folks we collaborated with from University of Leipzig. And Joe Blanti from, some of you may know, from Georgetown University. And essentially, they found the same thing. They also found that there was more calprotectin, and that this was happening in patients who were symptomatic and were exposed to foods they were reactive to. It was not happening in the control group that was not symptomatic. It was not happening in the food-sensitive group when they were exposed to foods that they were not ALCAT sensitive to. So the test was quite specific there, reinforcing that finding. This is just a summary of that. And a conclusion was that the induction of NETs that contain pro-inflammatory mitochondrial DNA may provide the critical link necessary for a better understanding of the pathogenesis of non-IgE mediated food allergy. And as you, I'm sure, have realized by this stage of the discussion, we're talking about non-IgE food allergy. We're talking about delayed food allergy or food sensitivities, not your classical type 1 allergy, like having an anaphylactic response to shrimp or so forth. So the ALCAT test, for those of you who don't know, is a whole blood test. It is not a serum test. And the advantage of this is that all the factors that could contribute to an adverse reaction that are in the blood, which means all the cellular elements, even the red blood cells, platelets, cytokines, antibodies, complement, and other proteins, they're all there. So it creates a situation which is a bit more in vivo-like than other approaches. So the main difference also is that it works. There's a high correlation with clinical symptoms, which has been established through double-blinded, placebo-controlled oral challenge. These studies are published and they're available in our dossier, which you can download from our website. And there's been a correlation with ALCAT responses to food additives of 96%. And those are double-blinded, placebo-controlled challenges. And of course, this is the only real way to determine if the food sensitivity test works. Because if you run a test on your patients and your patients get better, could they have gotten more improvement if the test was more sensitive? Could they have achieved the same outcome without having to eliminate as great a number of foods if the test was more specific? So you don't know that by just looking at an outcome. Well, the patient got better, so the test is great. Well, the only way you can really quantitate the efficacy of a test is to compare it against a known standard. And that's eating the food, but doing it under carefully controlled, rigorous conditions, which are double-blinded so that there's no conditioning or expectations associated with it. And doing the challenges in such a way that, you know, you do them, say, once a week, you introduce a new food, rather than every few hours or even every day, because there could be carryover from a previous challenge. So those kind of things are used by academicians. It's not practical for clinical use, but it is really useful for identifying or establishing the efficacy or lack of efficacy of an in vitro test for food sensitivity. So ALCAT's kind of unique in that regard. There's a significant improvement in metabolic health. There's a number of studies that demonstrate people lose weight and improve body composition without cutting calories, which again, inflammation impacts metabolism. I think we are aware of that right now. Improvement in all, for example, GI, neurological, fatigue, musculoskeletal, and so forth. And as we see, it identifies foods that are associated with cell-free DNA and formation of NETs. So just briefly, I don't want to get too technical, but an idea of how it works. So this graphic will, if I can get it to work, sorry, shows how we do this test. So essentially, whole blood comes into the lab, and then it's diluted in a neutral buffer, and it's put into it's aliquoted into a well, which has a test substance, a food extract, a chemical, an herb, or whatever, a drug. We're starting an evaluation. We've developed this test for the excipients that are used in the vaccines for COVID to see if we can identify adverse reactions to those components. We'll start that March 1st. And the way it does it is by actually looking at the cells one at a time when they're exposed to a substance. And the way that works is that the cells go through a sensing zone. So the cells that have been exposed to a substance go through a sensing zone, and they're recorded one by one. And then the aggregate of that data is then plotted onto a curve where we get a size distribution curve of the population of cells that we've just analyzed. And it looks like this. It's just sort of a standard histogram. And this would be the baseline. I didn't mention that we run a number of wells without a test reagent. And in the same way, we average those and get a baseline. And then we get a curve for each individual aliquot of blood that's been exposed to an individual substance. And then we compare the two. And if there's been a change, and this is kind of typical if there is a reaction, if there's no reaction, the two curves just match up. If there is a reaction, they don't match up. And there can be different sorts of ways in which they don't match up. In this way, we actually see that cells have gone away. A lot of them have swollen, and some of them have swollen to the point where they've actually burst. And of course, they release their contents and extrude their contents, which includes toxic mediators that are tightly stored in granules can get released, as well as free radicals, which are generated, and NETs that do what they do. Now, normally, the neutrophils, as I mentioned, are the most prevalent innate immune cell. The innate immune cells are the monocytes, macrophages, dendritic cells, and in the circulation are monocytes and neutrophils and basophiles and eosinophiles. 90% or 85% of these are the neutrophils, and they're relatively short-lived, unlike the lymphocytes, which might live for a year or so and contain memory. But that means about neutrophils, no one knows exactly how long they live, and they're very interesting. They might live 5 days or 10 days or 12 days, but about, it's estimated 100 billion of them die every day. So that's a lot of stuff that has to be cleaned up every day, approximately 100 billion under normal homeostatic conditions. So if you were to take the DNA, not just from the neutrophil, but all the cells in the body, all the cells, and stretch it out end to end, it's kind of a remarkable statistic. It would stretch from one end of the solar system to the other and back again. And each cell contains approximately 1 meter of DNA. So that's something that's not, it's kind of overlooked in functional medicine. Hey, homeostasis means, our hormones are in balance and so forth. And we know generally our immune system needs to be in balance, but there's a lot of other processes that are going on in a big way that have to be looked after. So people with homeostasis, they have enzymes to degrade this. The macrophages will eat up these things. And in fact, if the cell is not if the neutrophil is not in an activated state, it will actually bias the macrophages that eat them into an anti-inflammatory mode, will be an M1 expression. In fact, they're using apoptotic cells in cell therapies to dampen inflammation prior to things like transplant. However, if the cells are in an activated state, and if they're activated, there's going to be the kind of phenomenon that we looked at before, where we see that non-apoptotic or non-programmed cell death at normal, dying quietly with your friends around in your sleep. When the cells are in an activated state, and then of course they're dying, following a process of mitosis, they induce a pro-inflammatory bias to the macrophages. So again, another component of the problems with imbalance in the immune system are chronic activation of innate immunity due to environmental challenges and so forth. So ever since phagocytosis was first discovered a little over a century ago by Metchnikoff, Eli Metchnikoff, when he moved to France, they started calling him Eli. His name is Ilya. He was born in Russia. He was one of the first ones to really understand the beneficial role of probiotics. But since his time, it was understood that the efficient clearance by phagocytic cells of the dying cells was important for immune function, which we now have more insight into. And the process is that the cells are opsonized, the dead cells are opsonized, or the apoptotic cells are opsonized by something called milk fat globulin endothelial factor 8. And opsonized means it coats a part of it so that receptors in the cell can grab onto it and then absorb it and then digest it. And the cell, will find me signals and eat me signals that help this process along. However, another major step in this vicious cycle is that NETs actually inhibit efferocytosis by neutrophil elastase. And if you recall, this is something that's greatly increased when you have an ALCAT positive, ingestion of an ALCAT positive substance. It decreases the surface markers or surface molecules on the macrophage, which will bind to the necrotic or apoptotic cells. And that's what it looked like. Here we have a macrophage, and here's a cell producing nets, and they're inhibiting this process right here. So again, if you think in terms of not 100 billion, but hundreds of billions of cells undergoing necrosis or pyroptosis or netosis in response to something that someone might eat every day, and then that's compounded because it might be 10 things that they eat or contact every day. So you have so much of this DNA, which unexposed like that is quite toxic. And then you also add insult to injury, but not being able to break it down, that's not good. So when we get the analysis done, we put it into a format like this, which is kind of easy to read. And it just lists the most serious, less serious, and much less serious items that we found to be reactive, and then the items that are found to be non-reactive and safe. And then we take this data and we also provide an eating guide, and we can provide a little bit more details by rotating these non-reactive foods. And that's essentially what a test report looks like. And again, you'll be able to see this in our scientific dossier. There's about 50 published abstracts and papers over the years, looking at a number of different conditions. And some of these things are kind of a meta-analysis of all of those up at that time. It's a little bit more in the last couple of years, but 98% improvement with weight loss and GI issues is 87% and so forth. Migraine responds very well to food, respiratory disorders, and so forth. So it's what you'd expect to see. And again, an ALCAT graph matched with a bright field microscopy, you see a decrease in cells where the test curve is lower than the baseline. That's what it looks like under microscope degranulation. We see a swelling or shift to the right in the granulocyte fraction, and the cells are not degranulated fully. They're not too viable anymore. And when the graphs match up, you have a nice, happy looking neutrophil. What else? There's NETs, but there's also oxidative stress. Number of factors produce that. There's no one thing, it's all a gestalt. But those are big factors. Years ago, many years ago, I started to look into, because we do see that when neutrophils get activated, there is generation of free radicals. One of the more interesting books, I think, about how that relates to aging was written a number of years ago by a professor of immunology from UCLA, William Clark, and he talks about in detail what Denham Harmon propounded in 1956, the free radical theory of aging. And he talks about how these free radicals, they're necessary for defense, but they get out of hand, they cause problems, and no molecular species is immune to the damage. They can attack and deform protein molecules, disrupt structural complexes, again, and what's causing us to age? It's a big factor. Inhibit enzymes and can attack the nucleotide bases that make up both nuclear and mitochondrial DNA. And neutrophils purposely generate high levels of oxygen radicals. In addition, even prior to NETs, even NETs to a great extent depend on the generation of free radicals for them to form. So free radical medicine is very interesting, it's very complex, but it's a major factor in terms of aging and tissue damage, stemming primarily from overactivation of the innate immune system. I think just in the interest of time, I'm going to just give the highlight of this. It's a University of Rome study from many years ago on ALCAT, basically looking at the ability of the cells to respond to a stimuli after they've already responded to a test positive food. And essentially what it's showing is that they cannot. So the cells can become exhausted. So we can have a condition, and we see that with COVID, where the immune cells on the one hand are exhausted, and you have a lot of netosis and debris and so forth, and yet they're not able to defend the body. So why? I mean, that's what the ALCAT is for, essentially, in a nutshell. The theme here is that the cells are responsible for the tissue damage that we see in aging and inflammation and all the other factors. I'm about to discuss a few more. Why not avoid those things? It's easy. Metabolic syndrome, activation of innate immune system, causes pro-inflammatory cytokine production, which will suppress natural cortisone. We increase the cortisone initially to quell the inflammation, then it's depleted, and it also inhibits sex hormone production. Inflammation shortens telomeres, as we're probably well aware, and that has a lot of negative consequences. We see a preponderance of circulating neutrophils in the blood of patients suffering from Parkinson's and Alzheimer's. So again, oxidative stress is impacting the neurological system. MPO, myeloperoxidase from neutrophils, is found to be more accurate in predicting heart disease than LDL. Oxygen radicals are also involved in preeclampsia in women. They're also involved in male infertility. So they find greater amount of circulating neutrophils or active oxygen species in males who are infertile. It's no surprise. Again, another connection, connecting the dots. More environmental challenge, decrease in quality of the food supply, less nutrition, so less support for detoxification pathways, more activation, chronic activation of innate immunity. And we see that today, more infertility. So for now, that's all I wanted to say about the ALCAT test. And I want to switch over to another test, which is testing the specific immune cells, the lymphocytes. And of course, the lymphocytes are become engaged when and if the innate immune system cannot quell an attack or an infection. Then the specific immune cells get involved. So what's an illustration of that? For example, a neutrophil might digest a pathogen and destroy it internally with free radicals and toxins like elastase. But suppose it can't grab onto the pathogen because it has a polysaccharide coating. Well, then a specific antibody will be made to overcome that. And that's produced by the specific immune system. So there's a working coordination there. Specific immune cells produce antibodies that also neutralize, hopefully. And they also will directly kill the cells that have already been infected. But they're specific. Each type of lymphocyte will recognize only a certain kind of pathogen. So that's why it's called the adaptive immune system and the specific immune system. And whereas innate immune cells get programmed through epigenetic modifications, specific immune cells evolve to become more familiar with a pathogen over successive generations. And they call that antibody maturation. And that's brought about by changes in the genome as well, not just the epigenome, but also the genome. So there's a lot of differences there. And once you have exposure to a pathogen, those cells that recognize it will stay in the circulation as memory cells. And when they encounter that again, they will mount an attack much more quickly. So let me leave it there for a second, come back to that. But our cellular nutritional assays are based on looking at lymphocyte responses to neutrophils. And the thought came from an experiment, very famous experiment, done by George Beadle and Ed Tatum at the University of Chicago in 1941. They were studying cell growth by looking at a common bread mold, an orange mold common on bread, maybe not so common anymore due to preservatives, but called Neurosporosa casa. So they put those in a essential growth medium, and they saw that the cells would survive, the spores would survive. But then they irradiated the spores, and they were no longer able to grow. So they added back in different culture, different aliquots of that initial replicating spores in media, different nutrients. And they found that eventually adding through a lot of trial and error, arginine was able to restore growth. So they derived from that something that became an essential part of DNA dogma, that one gene encodes for one enzyme. So an example of that would be what Kilmer-McCulley wrote about in the 60s. Kilmer-McCulley from Harvard, who wrote a book called The Homocysteine Hypothesis, which talked about homocysteine being an intermediate, metabolic intermediate amino acid that was toxic and immunogenic, that if a person lacked certain enzyme efficiency, which was genetic, would accumulate too much of, and that would cause damage to the arteries and eventually lead to cardiovascular problems, more so than LDL. And that wasn't super popular with mainstream, and essentially he lost his job. But that led us now to routinely testing for methylene tetrahydride folate reductase, which is a key enzyme there, but also other enzymes are involved in that pathway. And we do that testing for this reason. And this is what essentially our report looks like. So it gives, again, these enzyme deficiencies, which are inherited, can be addressed with nutrition. Coming back to the lymphocytes, this is an old graphic, but it's very good for purposes of understanding. When we talk about specific, innate immune cells, they'll have direct contact with a pathogen or an incitant or a toxin or a sterilant. So the specific immune cells go through an intermediary almost always, and that's an antigen presenting cell, which will take up the pathogen, digest the peptides, combine it with a certain complex called an MHC complex, major histocompatibility complex 1, translocated to the surface of the cell, and then a T cell that recognizes it, and only the T cell recognizes it, and that's why there's specificity, will become activated by that primary signal to the T cell receptor and the secondary signal, which is not illustrated here. And then that cell, either if it'll be a memory cell that recognizes it, will then divide. And innate immune cells simply just don't do that. It will send signals to the B cells, and certain B cells that recognize that pathogen will become activated, and then will begin to secrete soluble forms of their receptors, which are called antibodies. Depending on which signal it is, will determine the type of antibody, which is very important because they have a lot of different functions. And then they start to multiply. So this is a classical pathway, well understood. And you can see that the ability of the cells to divide will determine how quickly we can quell that present challenge or infection. And of course, cells need nutrients to divide. So what we do is we take the cells, we put them into buffers with some essential growth substances, and the patient's own serum, because that's their normal condition. We stimulate them with a mitogen because we want to activate all of them, as opposed to just those few that might activate to a particular food or pathogen. So we use what's called a mitogen. Mitogen means mitosis generator. And then we add individually a number of different substances to each aliquot. And we look for changes in the metabolic activity of the cells. And if we see an enhancement of the metabolic activity of the cells, there's a reasonable inference that there is not optimal, regardless of the serum concentration, there's not an optimal level of that particular nutrient in the cells in order for the cells to function at their best. And then we report that out as being either sufficient or kind of borderline or insufficient. Then we do a variation of that where we stress the cells. And we look at how well cell survival is. We stress the cells with oxygen, free radicals, hydrogen peroxide. And then we do that by adding in substances and see how well we can inhibit that. And then report those out. So these are a number of antioxidants and botanicals that might be beneficial for any individual. So implementation of this nutritional aspect over and above what we know and do every day in terms of advising people on healthy lifestyle and prevention, which includes sleep, exercise, and everything else that has an impact on our epigenetics. We can use these tests a little bit or a lot to make it particular to the individual. Let's find, certain foods in general are not healthy, but some even healthy foods may be bad for some individual. And it's good to know what those are and what foods or nutrients will enhance immune function. So thank you very much. Whoops. We've covered this, and if there are any questions, if anyone's still here, glad to answer those.
Roger, thank you so much for that. That was amazing. We had a ton of comments inside the chat the whole time, speaking about how valuable this information was. And we had a ton of questions. So let's go ahead and just jump right in. And for those of you who do have to jump, it is 12 o'clock, so completely understand, no worries. We'll actually send out a recording of this session. So if you happen to hop off and miss the Q&A, no worries. We will send out a copy of this, but let's, again, just jump right in. So the first question we have, and just so the chat is aware, we're going to go chronologically here. So the first question that we got was on the gender study. So does this gender study account for where the female is in their monthly cycle, as well as in their life cycle, perimenopause versus postmenopause, as those would be factors?
Excellent question. I do not think so. I'd have to go back and check that, and references as whoever asked the question and check it also. I do not believe that to have been the case.
Great. Question number 2, what is the best way to explain to patients what is being tested on CSS food sensitivity test?
Simple kind of way to explain it is how their immune cells respond to the substance directly. Not whether there's an antibody or a protein there that may or may not, induce a cellular change is going to cause the release of free radicals and toxic mediators and possibly mitosis. But do your immune cells react to this? We're actually looking at the immune cells while the cells are still alive and viable in the context of the whole blood, which is more in vivo-like. So do the cells respond or do they not respond?
Yeah, absolutely. So question number 3, if a person has leaky gut, will there be more food IDs due to excess inflammation? Is it better to work on healing the GI as much as possible before doing the test?
Excellent question. Could do, but it's both, because the toxic mediators like elastase cause leaky gut, as well as other factors like cortisol and stress. So you might want to do the test initially and then repeat it sometime later, because even if you're working on healing the gut, which is a great thing to do, if you're still eating some foods that are releasing elastase and other things that cause leaky gut, you won't succeed. So you'll be caught So I think it's beneficial to use both protocols, dietary avoidance of susceptible foods and also healing the gut at the same time to support the natural healing process. The gut will heal if you take away the factors that are causing the local inflammation there.
Mm-hmm. Definitely. So question number 4 we got here. Can Roger talk about the upcoming beta test of the innate immune system as related to the vaccine? What do you expect to find? Why would the vaccine send the body into haywire and cause a return of an autoimmune disease that was in remission?
Wow, see, that's really complex. And one of the reasons we're doing this is because one of our close collaborators has been working with vaccine for decades and decades. He actually studied with Salk. And there's been a lot of confusion around this area of adverse reactions to vaccines. And, you know, obviously it's spurred a tremendous amount of controversy. And it's such a big issue today. So we don't know what we're going to find, but before I speak about what we're going to do, I just want to mention we're going to test for the excipients that are used in the vaccine. For example, there are a number of things that are used in the delivery system, these nanoparticles that encase the RNA. There are other things like polyethylene glycol and various polysorbates. So these factors have been shown to cause reactions in some people. However, when we're talking about things like autoimmunity induced by the vaccine, my own personal feeling, and I want to say this up front, is that that's probably, if that's happening, and I'm not doubting it, but I'm just cautiously stating if that's happening, it's probably more likely that it's happening in response to the actual spike protein itself, rather than the excipients. The excipients may be important for many people, and that's what we're going to test for, because that's all we can test for, because we can't test for the spike protein. We could if we had it, but I don't think there's any sense, because honestly, the purpose of it is to activate the immune system. So we're just going to look at the excipients. Now, it's no surprise that it could be that either exposure to the virus, either through the vaccine or otherwise, could induce aberrant immunological effects such as autoimmunity, because neutrophils, as we know, being active and releasing NETs as well as other factors, can initiate and progress autoimmunity. So what we expect to find based on preliminary evaluation is that some people will be reactive to some of the components used, the so-called non-active components, the excipients, and they can use that information however they want, but one way is to choose one vaccine over another.
Yeah, that's great. Appreciate you putting in all this work as well. This is amazing and so important. The next question would be, can this test differentiate between the same food or plant in different forms? Example, cooked beets versus raw beets, or plant in a certain dose, herb or concentrate or essential oil?
You know, it's going to be different for every person. So, and some of these things are dose dependent. Unlike let's call it a classical allergic reaction, where someone smells peanut, where they kissed their boyfriend who ate a peanut an hour ago, and they have a strong acute reaction, anaphylaxis, and they die. You know, a couple of molecules of the peanut allergen is sufficient to do that. Innate immune responses or sensitivities are dose dependent. So we try and calibrate the test so that we have a dose which is sufficient to demonstrate a cellular reaction ex vivo, which will correlate in vitro or in vivo, ex vivo versus in vivo. Does it matter? Yes. So if a person is reactive, let's just say to apples, if they have a bite of an apple, it might be fine. Or 5 bites of an apple might be okay. If they eat 3 apples, it's not okay. So it's a bit of a way of using that as a guide. It's not definitive. It's not giving you something super quantitative where you can say, I have this degree of reactivity. Cooked versus raw. We've looked at that carefully because there's a lot of discussion about that. It matters a lot with type 1 allergy. Again, because you're looking at proteins, like a protein, the peanut allergen, for example, crustacean allergen, these are proteins. When you're looking at a delay type sensitivity, it's usually not the proteins, it's more other chemicals. And I'll tell you why. You know, all plants and animals, particularly plants, produce toxins to protect themselves from insects. And if we've grown up with them, meaning we've had exposure for generations and generations, we have the enzymes necessary to break down those toxins. If they're somewhat novel and our genes don't produce the enzymes to break those down in our detox pathways, and or if detox pathways are compromised because they're overloaded or undernourished, then the innate immune cells come into place. So the differences between allergy and intolerance with cooked and raw is not such a big deal. So if someone comes up positive in the ALCAT test to apple, we're testing raw apples and we're using all organic stuff because we don't want to confound what we find, because we do then also test for preservatives and additives. Does it mean you shouldn't eat cooked? Yeah, probably. You could try it, but most likely, if you're allergic or reactive or sensitive to the raw form, you're probably going to react also because the chemicals are not so much changed by heating and cooking as are the proteins.
Perfect. Thank you for that. So next question would be, do we want more programmed cell death, i.e. apoptosis?
It happens, you know, apoptosis is normal. Cells get old and they undergo that. Now, are there compromises where autophagy or mitophagy is insufficient? Yes. So do we have senescent cells in the body that we want to get rid of? Yes. Do we want to enhance that? Yes. It's normal, but using so-called senolytics might help that. And I think that would be, I'm not an expert in that field, but from what I've looked at, senolytics have a benefit. We do want, not necessarily more, but we want appropriate, just like anything else, appropriate destruction of cells that have outlived their usefulness.
Yeah.
So I just want to point out, we don't want to cause the internal damage to the cells prematurely, so that has to happen. We want the cells to be happy in the first place, but if they're gonna die, and everything has a life, you know, we do want to get rid of them. We do want to clean those things up.
Yeah, absolutely. Makes sense. Last couple questions here. Is ozone therapy damaging to the body?
I don't know. I'm going to take a pass on that. I wish I knew the answer, but I just don't.
Yeah, no, appreciate that. And then final question we have here. And again, just to reiterate, the recording of this is going to be sent out. So if you do have to hop off, no worries at all. Final question, is the CNA just related to what nutrients will support immune function, or does it also relate to actual deficiencies?
I think they're related. I think the immune cells are like good cells to study because you can take them outside the body easily enough. You can't biopsy other cells so easily. And they are metabolically active cells. You're accelerating the processes. And especially when we stimulate the cells with the mitogen to become active, I think they are reflective of the nutritional status of other somatic cells.
Perfect. Well, Roger, thank you so much for joining us this afternoon. It's been so insightful, and I know the audience has absolutely loved it. You know, with that, is there any last thoughts or anything you want to get out there?
No, I just really want to say that, you know, it's a pleasure to see people interested in this topic. We're continuing our work in the field. Our company's name is Cell Science Systems. We're actively working on, we've actually brought out a couple of more tests that are cell-based tests looking at cell function and looking at free radical generation and looking at which cell subtypes are involved in these reactions so that we can better understand them, because there are so many complex health issues these days and medical issues and diseases that are related to dysfunctional immunity. And that it's a super exciting area to be involved with. Of course, there's other areas that we're totally fascinated with and are equally valid and important. But this is a new area. There's a lot, you know, ALCAT, to be honest, I've been doing this for a long time. It's still sort of disruptive. I'm not sure why. So, you know, stay tuned. We're going to keep at this, and we've got a few more cellular assays, functional cellular assays that will be beta testing pretty soon as well.
Yeah, absolutely. Well, thank you so much again for your time this afternoon. We're super excited to have you here and looking forward to having you back in the future as a recurring guest with us. But with that, y'all, I'm going to hop in and I'm going to move over to chat a little bit about Rupa Health and show you exactly how you can order this test that Roger just went over. So for those of you who are unfamiliar with Rupa and have maybe 10, 15 minutes to spare, please stick around, and I'm going to go ahead and jump right in to chat about Rupa Health, who we are, what we do, and how you can actually order these tests that we were chatting about today. So with that, y'all, my name is Adrian Martinez. You've probably seen me hosting this Q&A as well as during that intro. And if you're not familiar with who I am the head of practitioner partnerships here at Rupa Health. What does that mean? Essentially, it's a very long title. That means that I chat to our practitioners on a daily basis about Rupa, who we are, answering questions to essentially set you up for success. So who is Rupa Health? Well, we're a platform that is designed to alleviate a lot of the pain points that can oftentimes be associated with ordering lab work. Most practitioners that I speak with order anywhere from 3 to 6, sometimes more. I mean, I've spoken with practitioners who've ordered 15 plus different labs at any given time, right? And that's a lot to manage. Not only do you have to set up separate accounts for all these different labs, but of course you have to hop in to each individual portal to track things such as your results, as well as placing the orders. And that's not even just mentioning one of the biggest pain points that I hear, and that's just the management of the patient experience. There's a lot of necessary, I don't want to call them evils, but a lot of necessary steps involved with these tests. And unfortunately, those steps can take a lot of time out of your day, out of your practice's day. If you're lucky enough to have an assistant, they might manage it. But if you're a solo practitioner, which a lot of our users are, that's time out of your day, which you could be better spent building your business. So how do we make things more streamlined for you? Well, the first thing that we've done, and again, I'm on rupahealth.com, is we brought on 25 plus labs onto our platform, allowing you as a practitioner to place an order from any one of our labs within a matter of seconds. Now, of course, this is dependent on your license type. So if you are able to order directly from those labs with your licenses, then you are able to order from Rupa Health as well. Now, if you are a practitioner whose licenses may be limiting in your ability to order from certain labs, no worries, we have a program for that as well that we'll be releasing here in the coming months. But what I'm showing you right now is the lab test catalog. As you can see, 25 plus labs here, over 2,000 different tests, of course, including cell science systems, which Roger just chatted about here. So you can order it all in one place. Beyond that, you can track and manage all of your results without having to go to each individual portal, right? The other component to Rupa Health, which is extremely valuable, is the patient experience, which I just mentioned a moment ago. So as soon as you place that order on Rupa Health, we can effectively take it from there. Whether it's managing billing directly with the patient so you don't have to manage any billing, we can do that for you, or we offer the option of you being able to pay for the test yourself and bill the patient outside of our platform. So it's flexible. We'll send over our own curated FAQs, instructions, oftentimes videos on how to take the tests, right? We'll walk the patient through the test taking process, answering any questions that they might have, managing any admin work, right, that can be associated with these tests, even coordinating phlebotomies, right? I know that's something that is big within especially the functional medicine industry is, hey, how do we coordinate these phlebotomies more effectively, more efficiently, so I don't have to spend hours, especially if I'm working within telehealth, to be able to coordinate this phlebotomy. We'll help manage that directly with the patient, and even up into including specimen issues. So if there is anything that goes wrong with the specimen, we'll be able to coordinate that directly with the lab and the patient, saving you time and energy when you would generally have to manage that process yourself. From there, you're alerted as the results come in, and you're able to view everything right within Rupa Health, making it as streamlined and easy as possible for you to track and manage all of your lab work. Now, that was a bit of an overview, but what does it all look like? Let's take a look, and I'll show you exactly really how simple it is to place an order on Rupa Health. All we need are 3 bits of information. We just need the patient's first name, last name, and email address to place an order on our platform. We collect everything else directly from the patient and make things more streamlined for you to place your orders, as well as ensures the accuracy of the patient's information should it change. What you're seeing now is the orders screen. So within the orders screen, you'll see at the top here that we can actually create custom bundles. A custom bundle being a set of tests, a set of blood panels, a combo of blood panels and tests from any one of the labs that you want to order from. It can be a combination. So that way it's just one click, and all of the tests you're looking to add in that bundle will be added into your cart straight away. Below that, you have the ability to create a favorites list. So a favorites list being a set of individual tests that you're commonly ordering. Again, with the idea here is to drive efficiency. You don't have to search through an entire catalog or log on to multiple different portals to get all your tests. They're right here in front of me. So if I want to order this ALCAT test 200 food panel from Cell Science, I have it right here at the top, right? But also maybe I want to order a GI map, right? So I have all my GI straight away from right here. And I can just order that GI map from Diagnostic Solutions. And there we go, it's right at my fingertips. And maybe I also want to order a mycotox, right? So again, just really showing you how simple it is to place these orders here on Rupa Health. And if you are looking for a test that is outside of maybe a bundle or your favorites, you have access to that entire catalog, which I showed you previously, right down below, the whole list of 2,000 plus tests at your fingertips. So once you add those tests into your cart, it can be as simple as clicking send to patient. We'll drop ship the kits directly to the patient, manage any details, any instructions, FAQs, support, right? But of course, there is some customization that we can do here. For example, we can schedule this order out. So if you are seeing a patient, and let's say that maybe you want to retest them 6 months down the line, as opposed to having to add this into your calendar to manually remember that, we can automate this process. You can schedule this out in advance, and that way it'll send the test to the patients straight away. You don't have to hop in and remember to send these tests to the patients, right? Below that, if there's add-on tests available, we have that available as well. So let's say with the GI, I want to add some zonulin in. I'll be able to click that button. It'll bring up all the details of the tests, breaking down things such as the sample type, turnaround times. I can download a copy of the instructions, which we'll send to the patient. I can view a sample report. If there are any biomarkers, we can expand this out and get an idea of all the biomarkers. But if I want to add that zonulin, again, it's just straight away as simple as clicking that button that we have right here. It'll default to having the patient pay for the order as well. So that's something that'll be really nice. I would say the majority, 9 out of 10, as an estimate of our practitioners prefer this method. And the reason being, well, there's a number of reasons. You simply just don't have to manage billing anymore, right? One less thing for you to have to manage. It makes it easier. But you do have the option, as I mentioned, to pay for the order. You'd simply just click that box. You'd add your payment information. It'll save it to the system. You don't have to add it every single time. And from there, you'd be able to bill the patient outside of our platform. I'll show you what that looks like in terms of communications to the patient here in a moment. You can add notes to the patient as well. So let's say that your patient as a supplement regimen that you want them to continue to take, right? You assume that there's probably going to be questions involved. You can actually add those notes here, go directly to the patient. Or if you have just general instructions that you're continually telling your patients to follow, you can actually add those in here as well as a snippet. So essentially a template, so you don't have to rewrite the same instructions over and over again. Again, efficiency here, right? That's kind of the key. You can add notes for us, and then you can actually add ICD-10 codes as well. So what will happen in this instance, you'll add the ICD-10 codes. Rupa will go ahead and send a template to your patient to walk them through how to create that super bill to submit to insurance for reimbursement. So that's how that would work. If nothing else, it's as simple as clicking send a patient. So that's how you place your order on Rupa Health in a matter of seconds from 25 plus labs, all at your fingertips. But I'm sure a big question that a lot of you have is regarding cost. How does our pricing work? What are the costs of the tests? Happy to answer that. So the way that our pricing works is very straightforward. We offer wholesale practitioner rates. And what that means is the same prices that you would get having an account directly with any of the labs will be the same prices that you'll see on our platform as well. So if you have an account with Diagnostic Solutions, you as a practitioner will get this test for $287.20. If you were to order direct from Cell Science, you'll get this test for $375, right? You're getting the same prices. The way that we generate our revenue here at Rupa is very straightforward. There is a 7% processing and ordering fee on each order. You'll see here, we'll break it down line item by line item, and this is paid for by whoever's paying for the tests. So what I mean by that is if you're having us bill the patient directly, then the patient will be the one absorbing this cost. Rupa Health as a platform is free to sign up for. There's no subscription cost. There's no monthly fee that you're paying. This 7% is the only way that we're generating our revenue. So if you're having us send these orders to the patients to pay directly every single time, then Rupa will be free for you as a practitioner. Simple as that. But where is everything tracked, right? Once I send that order to the patient, what's it look like on my end as a practitioner? Well, within this main screen here, this is where you'll track all your orders. You're able to search by patient. You can filter by status of your orders. So we'll consistently update the status of your orders along the way. So you don't have to log into each portal to view the status of all your orders. And you can click right into them. So as you can see here, this is an existing order that I have out. I'm able to view when the samples arrived at the lab and when can I expect the results to come in. Simple as that, right? So that way I can know my timing. I can plan accordingly with my patient. And as the results are in, I'm able to hop in here and I can view those results directly within Rupa Health. So I can download them straight away. These are the same exact results that you would be receiving directly from the labs. Rupa's not making any interpretations or many adjustments. These are the same exact results. You can send the results directly to the patients from our system. This is completely HIPAA compliant. We will not send the results to the patient without your consent. So you have full control over this. You can even schedule a clinical consultation. So should you need some assistance interpreting the results, you can schedule a consultation directly with the lab and schedule some time with somebody on their staff to go over the results with that interpretation. And then finally, you'll have access to that digital requisition. So what I've shown you so far is how to place your orders on Rupa Health, how to track and manage all of your results. But what does the patient see? Let's jump right into that. So this is the timeline of what the patient experiences on Rupa as soon as you place that order. So your patient will be notified once you place that order. The kits will be shipped out within 24 hours of payment. So something to note here that makes us a little bit different than working directly with some of our lab partners is that Rupa will wait till payment's received before sending the kits out, okay? We'll send the FAQs, instructions, videos to your patient within 48 hours. And then we'll follow up with the patient to ensure that the process is going well, answer any questions, and they can reach out to us at any time. And then finally, you are notified as the results come in. So what does that all look like? Well, this is the email that your patient will receive once you place that order. Hi, Joshua. Dr. Jordan has ordered these tests for you. We will introduce who we are, and then we'll highlight the different payment options that we accept. So really big call out here. Not only can we accept cash and credit, but we can also accept HSA, FSA, and we can even set up a 3-month interest-free payment plan directly with your patients. One of the ways that we're helping to lower the barrier of entry to get access to these very oftentimes expensive tests, but very important tests, right? And so we will go ahead and provide these additional options, and we'll even send out the kits after the first installment of that 3-month payment plan. We're not waiting for the entirety of the payments to come through. we got it. We'll send it out after one payment, and your patients will be ready to rock right after that. From there, we'll collect all the necessary information to complete the order, billing information, shipping information, demographic information, and then we will highlight the cost of the tests and break down everything very transparently. Should you decide to pay for the tests, nothing changes in terms of the patient experience. We will still manage that end to end. So don't worry about that, but there will be some key differences in the communications. Well, really just one key difference. We're not going to show the cost, right? So if you as a practitioner are paying for the tests and managing billing outside of Rupa, we're not going to show the patient how much your tests cost and how much they paid, or how much you paid rather. And we're not going to collect billing information, of course. We'll notify the patient as the next step once the kits have been shipped out. And from there, we'll send over an email that looks similar to this. So we will send over instructions for each of the tests, FAQs, oftentimes videos. We will walk them through how to fill out their requisition forms. And as well, if there is a phlebotomy required, we can manage that in a few different ways. We can either customize the instructions based on your recommendation. If you are lucky enough to have a preferred phlebotomist or one in-house, we can take that information and send it to the patient. Otherwise, we will send over the options based on the lab that they're working with. And if the patient has any questions, whether it's, hey, are you able to send over some additional options, maybe a mobile phlebotomist option in my area, or have questions regarding the test, no worries. We will be able to answer those questions on your behalf and facilitate the whole support experience for you and for your patient. Now, of course, if it's anything medical related, we will send them back your way. We want to keep you in the loop the entire time, but anything support related regarding just the testing experience, we got you there. You'll be able to see an example on the right-hand side of some of our test instructions. This one is the Dutch Complete. As you can see, very comprehensive, but more importantly, in my opinion, it's very user-friendly. You're able to click through to each different section, see what's important, key details, really basically anything that they might need. But just to reiterate, if they have any questions, they can reach out to us and our team. We actually prefer them to reach out to us or our team, so you as a practitioner don't get tied up in managing all that support. Follow-up. Underrated. I'll just say that. I think following up with patients is something that can easily, kind of fall by the wayside, especially if you're busy, right? But they're so important to ensure compliance with these tests. And we've automated this process. We've automated a lot of processes, honestly, to ensure compliance and really get your patients to complete the tests. With our technology, what we've seen, our compliance rates up above 85%. And so very strong compliance rates that we're seeing. And so with that, you're notified as the results come in. And again, you're able to view them all within Rupa Health. So 3 core factors that you've seen thus far with Rupa Health: how to place your orders, how to track and manage your results, and then how to have your patient experience managed. What we're doing is we're providing an experience, we're providing a technology to provide that experience that's come to expect it in 2022, right? But we're more than just a platform to be able to order all your results. And I use that term platform because you're all here on our platform today, right? Rupa University is the class that you're currently all in. We host these on a weekly basis. So, Roger was gracious enough to join us today, but we host these on a weekly basis. So if you are interested in continuing to learn and continuing to grow, join us every week. I host these classes generally on Wednesdays at 11 a.m. Pacific, 2 p.m. Eastern. And so we host with doctors and influencers and practitioners within the functional medicine landscape who come and give these hour-long sessions every week. They're free. They are not CE, so keep that in mind, but they are interesting. And they'll always be talking about the different tests. So some amazing content here. And even beyond that, we have a lot more, right? We offer not just these live classes, but we have a magazine, a podcast. All of this is part of Rupa Health and really just kind of the functional medicine community that we're looking to build here. So with that, y'all, I'm not going to take up too much more of your time, but if you are interested in joining Rupa Health, rupahealth.com, it's free to sign up for. And if you have any questions, don't hesitate to reach out to me. This is my contact information, and I'm more than happy to schedule a call to chat about your practice, whether you're a solo practitioner, whether you're a large multi-practitioner clinic. We can work with you. We have a number of different customizations that we can do with your account. So if you have any questions that I wasn't able to cover during this presentation, please reach out. Let me know if there's anything that I can help with. But thank you again all for joining me this afternoon. It's been a pleasure, and we're super excited to share this content out with you here within the next few days. Look out for an email from me following up, and then another email within the coming days of a recording of the session.